you were part of a recent great paper just out in Cancer Discovery, and it was based on the JAVELIN trial, so axitinib and avelumab phase 3 trial. So really a great effort, more than 880, I think it was 886 or so patients. And you all not only did transcriptomics, but also had this nice correlatives kind of peripheral blood treatment over time, changes. And so I wonder if you could just kind of give us, what were the big questions going into this trial from a translational correlative perspective? And then maybe if you could highlight some of the big findings that you came out with.
Yeah, absolutely. And I think looking at the background, so this was the phase 3 study of avelumab plus axitinib, so an anti-PD-L1 plus a VEGF TKI versus a TKI alone. And this is what ultimately led to the approval of avelumab plus axitinib in that first-line setting for metastatic clear cell. And there was a wonderful biomarker paper led by Dr. Motzer and colleagues, Dr. Schwery and others, in Nature Medicine about, 4 years ago now almost, in 2020, where it was the sort of first look largely at tumor intrinsic things, but beginning to look at some host factors as well. And there were some key findings, I think, from that paper. Their study, much like our prior work, had shown that total mutation burden, while predictive for anti-PD-1 response or anti-PD-L1 response in other tumor types, didn't really have the association in kidney cancer. They identified an immune signature, largely, I think, T-cell mediated, but a general immune signature that really predicted for benefit from the IO-containing arm, and then had an angiogenic signature that really seemed like it predicted for benefit from the sunitinib arm. And so those were different, but I think parallels to what had been described by David McDermott and others in the Motion 150 trial, but I think really sort of hammered in a phase 3 setting that these tumor phenotypes actually make a difference in terms of response. And then it also found that individual mutations, in particular what they call double mutants, where you had mutations in 2 of a handful of genes that seemed like they each individually affected progression-free survival, really did seem to stratify patients by progression-free survival. And so that was really the initial foray into biomarker discovery work based on this JAVELIN Renal 101 trial. And really, I think the motivation for this second work was to move away from just tumor intrinsic to really looking at circulating biomarkers as well. And I think a lot of the work I've done in the past as well, and our group has worked on, has really been tumor focused and tumor microenvironment focused. And I think that's really important. But I think sort of the lessons learned over the last few years is it's probably not going to be a lot of low-hanging fruit for biomarker discovery, for really effective biomarker discovery, that just picking one gene at a time and seeing if it's mutated or not, that's not going to be the holy grail for deciding whether therapy is going to be beneficial in the way it is for EGFR mutations in lung cancer. And so I think what's ultimately going to be needed is to sort of lean into the complexity and really integrate many factors together, what's going on in the tumor, what's happening in the host, and what's going on in the peripheral immune system as well. And I think this paper really sort of sets a foundation to try to do that. And so the idea is to really do longitudinal blood sampling and an analysis of baseline tumor to really try to understand, first of all, how things might evolve with either sunitinib or avelumab plus axitinib, but ultimately how these factors might individually or together sort of impact response to therapy. And so a number of parameters were looked at, first kind of within the peripheral blood, things that affected sort of both treatment arms, which were longer progression-free survival in patients with lower platelets, lower myeloid populations, particularly granulocyte populations like neutrophils, and high lymphocytes. And this is something that I think is more reaffirming than anything else. We know that having a lower neutrophils and higher lymphocytes is generally prognostically good. And I think one of the things the IMDC really hints at is those patients with IMDC poor risk disease really have a sort of systemically inflamed state. And I think that's often reflected by, as we know, thrombocytosis, neutrophilia, and these sorts of things. So I think a good sort of reaffirmation there. I think what's interesting is that while sunitinib really did seem to affect the levels of various immune populations in the periphery, that you really saw these oscillations with the cycles of sunitinib, with avelumab plus axitinib, things remained relatively constant over time. And so I think that's helpful to sort of understand the impact of these therapies on sort of immune cell dynamics over time. The next big analysis was really looking at the T cell repertoire. And so there's a couple things you can glean from that, both within the tumor and the blood. You can use it as sort of a way to infer what is the total number of T cells. That's certainly something one can do. And then also how polyclonal or oligoclonal they are. Essentially, do you have one T cell population that really seems like it's expanding a lot, or do you have a really diverse range of T cells that none of them are really expanded, but there's a lot of distinct ones that are seen as well. And so within the blood, there was, within the sunitinib arm, a higher number of T cells actually associated with a longer progression-free survival, better progression-free survival. But we saw that the immune therapy, perhaps as expected, led to greater changes in that T cell receptor repertoire that really led to higher clonality than with sunitinib. Within the tumor itself, we saw, I think perhaps unsurprisingly, that higher T cells, sort of inferred through this method, was associated with a better progression-free survival with the immune therapy. And I think that really mirrors what was seen before, that Javelin immunosignature being high, being associated with better progression-free survival, very sort of similar findings here. The next set of analyses, I think, really gets at the heart of it, which is really trying to integrate these factors together. Tumor genomics and host genetics on the one side, circulating biomarkers, and then tumor microenvironment. And so it really looked at these double mutant population, which was defined in the original paper and refined a little bit here further, which included somatic mutations in 6 genes and germline polymorphisms in 4 genes. And what was observed when you stratified, so we knew from the prior work that the double mutants, tumors with this double mutant phenotype, had better progression-free survival. But now we could begin to look at, well, within those individual groups, either the wild type or single mutated group or the double mutant, were there any additional predictors? And what was interesting is within the wild type or single mutant, having more T cells was helpful, having activated NK cells, things we typically associate with effective anti-tumor immunity. But within that double mutant group, it was actually the presence of memory B cells, high memory B cells and low naive B cells, that was really associated with better outcomes. And so at least hints at the idea that there might be something beyond a pure T cell mediated, NK cell mediated killing that could take place there. Those double mutants also had higher proportions of follicular helper T cells. And so again, raising at least the possibility that things like tertiary lymphoid structures and B-cell mediated immunity might have some role. And I think that mirrors what other groups have seen as well. Work from the Friedman group using spatial transcriptomics had shown that tertiary lymphoid structures that produce immunoglobulins and actually coat the tumor really seem to be associated with better outcomes with immunotherapy. And our group has some work in progress where we similarly see tertiary lymphoid structures associated with good responses, particularly exceptional responses to immune therapy. And so I think we're really seeing that particularly in this double mutant group. And then what we also saw, connecting it with circulating cytokines, is that the double mutant group had lower cytokine levels for things that tend to promote myeloid recruitment and development, things like eotaxin-1 and MCP-1 and MIP-1 beta, things that are really myeloid recruitment chemokines in support of myeloid development. And sort of in line with what you might expect, if that double mutant group had lower myeloid chemokines, they also had lower actual levels of those myeloid cells and granulocyte cells like neutrophils. And so I think overall what the study sort of, I think, makes an initial attempt to do is to move a little bit away from the sort of looking at one factor at a time that certainly our group has done extensively, and beginning to really get the idea of integration, and looking at all of those factors, tumor genetics, microenvironment, circulating immunity, host genetics, all of those together. I think this is a first step. There's a long way to go, but I think even with this early work, seeing how these things really do link together, and I think it's going to be crucial to understand the integrated markers in order to have a better predictive ability.